Hello, good morning, everybody. I'm Carlos Teixeira. Welcome to the second day of our Fortaleza Austral Spring School. Thank you for being here again. Today is our second day, so I'm sure we're going to have very good talks and very good discussions like yesterday. And today we're going to have two sessions about conservation and management. I'm going to be the chair of this morning sections, and we're going to have three presentations. The first one is from Professor Tito Lotufo. Professor Tito Lotufo is a biologist and has a PhD in biology and zoology. He used to work here with us in Ceará, but a few years ago, he moved to the Oceanography Institute of the University of São Paulo in Brazil. And Tito's research deals with zoology and reefs ecology. And he also works with conservation of reefs. So, Tito, thank you for being here with us. It's a pleasure to have you here, and you can start your presentation.
Oh, thank you very much, Carlos. I would like to thank also the organizing committee for this opportunity to talk about some of my work. So I'll start my presentation right now. Well, in the next few minutes, I intend to talk about the hidden creatures that live in the reefs and how we can monitor this fraction of the reef biota. So, as Carlos said, I'm now coordinating the Reef Biology Lab in the Oceanographic Institute, University of São Paulo, here in Brazil. So, what is the cryptic fauna, as we call it, cryptofauna of the coelobiotes? Well, this is the smallest fraction of animals that inhabit the reefs, and they live on crevices and spaces within the reef matrix, or in spaces within the arms and branches of the corals and other organisms in the reef. So in this cryptofauna, we can find virtually every group of animals. So it's a very diverse fauna that lives within the reef matrix and associated with the reef. As here in this picture, you can see some examples of the cryptic animals that we can find. These are some of the taxa that lives within the reefs, so like sponges and cnidarians of different groups of cnidarians, and not only the big corals that we see that are not cryptic, but many hydroids and other cnidarians live within the reef matrix. There are many groups of worms like flatworms and different groups of mollusks from bivalves and gastropods and the beautiful nudibranchs, and many species of annelids, like polychaetes, that live within the reefs. And one of the most diverse groups in the cryptic fauna are the arthropods, like the crustaceans, small crustaceans that like crabs and other groups of crustaceans that live within the dead coral and all the material that makes the coral reef matrix. There are many bryozoans as well. Ascidians is also an important group, and also, of course, vertebrates like small fish, cryptic fish that may lay eggs in the crevices and in the hidden spaces within the reefs. So it's a very diverse group of organisms that occupy these spaces within the reef matrix. So why we should worry about the cryptic organisms in the reef? Well, they comprise sometimes 2/3 of the reef biodiversity. So most of the reef biodiversity is actually hidden within the reef. It's not on the surface. It's not part of the big corals and fish that we can promptly see when we are diving or we are watching a movie about the coral reefs. So most of the diversity is hidden within the reefs, like the inhabitants in a big city. So if you look from the outside, you can see the buildings, you can see everything, but you cannot see the people inside the buildings. So, and this cryptic fauna, these cryptic organisms, they can perform many important functions in the ecosystem. Many roles in the trophodynamics, so they take part on many of the links for energy and matter transfer within different compartments and different groups of organisms in the ecosystem. They are important carbon sinks, so most of the biomass may also be on this component, in this cryptic component. They are important also in the bioerosion of the reefs. So this is part of the balance on the coral reef between the reef building and the bioerosion processes that are a constant part of the dynamics of the coral reefs. They are important also in binding and stabilizing the reef matrix. So they bind, they glue together all the pieces and sediment that fall within the crevices, so they incorporate this part of the reef, and they are important also in the diagenesis, or the process of building the foundation of the coral, the actual organic part of the rock that are being formed and part of the coral reefs. And they play many different relationships with other organisms. So they may be parasites, they may be in symbiotic and mutualistic relationships. And this is a very well established for some of the organisms, but there are many things that we don't really know about the cryptic fauna. Just an example, I can show you here a paper published by Seabird McCann and Jenna Moore, talking about the importance of these small crabs, the trapezia, for as guards in coral reefs. So they defend the corals against, especially the attack by the crown of spines starfish that can really be devastating in the coral reef. So they eat all the coral. They may be large populations that wander and start eating all the coral. So these crabs, they are very effective in defending and fending off the sea stars. So here we can see if the crabs are removed, the tissue consumed by some other, in this case is a gastropod, is much larger. So the crab can defend the corals against the predation by many different organisms. So this is just one example. There are many others. And they're still understudied, especially because they are concealed within the reef matrix. So they are really hard to collect. They are very small. The taxonomy of these small groups is complicated. It's very complex. Not many taxonomies will tackle these problems of taxonomy of small organisms that depend on microscopy or special procedures for you to really identify them. So this is a big problem, and that's why they're still not properly understood. There are many efforts in the last years to beginning to, at least to inventory this component. So there are many expeditions from different organizations around the globe, and just when some as examples, like an expedition to Kumak conducted by Philippe Boucher from the Muséum National d'Histoire Naturelle in Paris, and a national effort here in Brazil, the ProBio project from the Environment Ministry, and also, of course, the Sense of Marine Life with different initiatives like the sea reefs that lasted from 2000 to 2010 in order to really understand the biodiversity. So during this time, in the last decades, many different methods were developed to start to really assess this component of the diversity in coral reefs. So here is an example of the narrow lift system where you actually vacuum the crevices and the spaces in the reef matrix in order to take out the organisms that are there. So it's like vacuuming the sea bottom. Also, different tools like Yabby pumps to get shrimps and other organisms that live on burrows in around the reefs in the sand. Also, reef brushing is another strategy to get small organisms, especially mollusks and crustaceans. So you actually brush the reef in the small net, and you capture all the organisms that fall from the corals and the algae and all the other organisms you're brushing. You also, of course, you hand collect many organisms directly, hand picking and taking pictures of everything. And there are all other more, a little bit more restrictive methods to collect these organisms, like the lumen, which is a net that is deployed on the sea bottom. They use it for shell collectors to collect seashells from the bottom, so they leave the lumen sitting in the bottom for quite some time, and then they get it back, and the organisms are all living within the or entangled in the net, so they get the net and start to process this on board. This is another very effective method to collect some of the mollusks. So just an example, the effort conducted by Dr. Philippe Boucher in Comac, on his first trip there, he was able, using different approaches that were not being used so far back then, he was able to collect 2,738 species, only in a single location around Comac in New Caledonia. If you compare to numbers from other places, like even large provinces like New Zealand, the Mediterranean, you see that this number is astonishing. So this is how we see that most of the diversity is still not being properly assessed, or it's hidden in the reefs. So there are many challenges when you intend to monitor or study the cryptic fauna. And the challenges are related to access and logistics. You saw that this is our organisms that live inside the reef matrix. So on a first approach, you may think of just getting blocks of coral, breaking everything apart and detecting what is there. So this is a very destructive method. It's not adequate for monitoring, for long-term monitoring, for instance. Also, you have to standardize the methods. There are not so much knowledge about the species that are within this component. It's very time-consuming if you think of, removing blocks or working on very small animals that you have to do the proper taxonomy and send it over to different groups of taxonomy. So it turns all this process very expensive if you think on a first glance, on a first approach. So colleagues started to figure ways of doing this in a more inexpensive and effective and standardized way. So the first approach was developed by, one of the first actually was proposed by Todd Zimmerman and Joe Martin. So they proposed this structure made of concrete, very heavy, but standardized. You can replicate this in different places to study the cryptic fauna from the reefs. And using this as a starting point, Rusty Brainerd from NOAA in the Pacific, the Pacific Island Fisheries and Science Center, and also Nancy Knowlton, which is now on the Smithsonian Institute, they developed a different tool based on what Martin and Zimmerman had done before. So they figured, they started to design a smaller unit made of plastic, of PVC, and then they first deployed this in the Hawaii in 2006. And since then, they have been used in very different places. There are other approaches for studying these cryptic organisms that may be associated, like the carbonate accretion units, where you deploy this on the reefs. There are small sandwich of plates that are left there, but the purpose of the cowls is to assess the carbonate accretion. So it's a destructive way of analyzing this after the deployment time. But you can also use it to study the cryptic fauna. But the arms is the topic that I want to develop more, but I've been used this for quite some time now, and I'm also part of the Global Arms program. So ARMS stands for Autonomous Reef Monitoring Structures. So since Martin and Zimmerman proposed that concrete arms unit, it was slowly changing into this new model of arms, which is composed now of 9 PVC plates made of gray PVC plates with spacers within them, and also attached to a base plate, and they are deployed in the coral reefs and on different systems, attached to the bottom by different things. You can use like steel rods or weights on the bottom to attach it to the bottom. And they stayed submerged in the bottom for 1 year up to 3 years or more, depending on where you're working. And you usually deploy them in sets of 3. So there you have also replicas on the same sites. And all this method was conceived to use not the regular taxonomic approach where you also take apart everything and examine all the organisms there, but it was conceived for use the metagenomic approaches. So it's intended to be fast and standardized, as I said before. But in order to do that, you have to be able to use a very good database on the barcodes of many different groups of organisms. And today, this map here shows a little bit of, it's not really updated, but it shows that arms are being used everywhere in the world, from the tropical to the temperate seas, and even in Antarctica, they are deploying arms now, and from the shallow reefs down to the deep reefs, deep reefs and mesophotic reefs, and even on the deep sea using ROVs to deploy these units. So there are just small condos that are left down in the bottom. Here is the page of the Ocean Arms program. It's coordinated by the Smithsonian, and now the global coordinator is Chris Meyer, which is the head of this project now. And on this website, you can find all the protocols, all the projects that are there, and arms that have been deployed. So it's a good start if you intend to be part of this initiative as well. So what happens when you deploy the arms and you leave them cooking in the bottom? After just a few months, you can see that it's been heavily recruited. It's like a condo for marine organisms, so they will get into these spaces within these plates and start recruiting. And then after 1 year, you go back and you take them out and you replace with a new unit, and you take this to the lab. So in order to go to the lab, you have to put a cover on it. So you cover the unit with a bucket or with a mesh, and put some rubber bands in order to take it to put it in place, and then you remove it from the bottom. So all the organisms, the motile and the sessile organisms, the cryptic organisms, are all trapped inside this unit. So you take it up to the boat and back to the lab. It will be sitting on a container with seawater all the time. And the lab is also totally set up for processing the units. All the method was conceived to be easy and simple, and it can be done anywhere with minimum equipment. You don't need like fancy equipment or things that are hard to find in order to process the arms on the field. So it can be done in the field. You can just, this is not a field lab, of course, but just an example. You can see what we use. There are trays and tubes. We have to bring all these large containers for keeping the arms there. So we have this all set up. And here is an example of the arms also already disassembled with all the plates sitting on the container, waiting to be processed. So once you start processing, what we do with them, we first of all, we take the water from the container and we sieve all this water to get the motile fractions. So this motile fraction, the big ones, are counted and identified right there on the spot. And the smaller fractions, they are all sieved and they're packed and they are reserved for metabarcoding. Then the plates are photographed with high resolution photographs in this setting. So we take like 15 pictures of each plate in high resolution, then they are stitched together in order to make a single picture in higher resolution like this one. So you can see all the sessile organisms in the plates. Then we can use these pictures in order to quantify what is there. And in the first approach, we still need taxonomists to be able to collect some specimens from these plates and also from the motile fraction and identify them and to generate the reference barcodes in order to do the metabarcoding later. So when you start doing arms, you have to worry about doing a proper taxonomy in the beginning, but this is only the first step. Then what you do, you get all these fractions. So the motile fractions are all get together, you weight them, and you put them on tubes with ethanol in order to process them in the lab later. And the sessile fraction and the plates, they are all scraped and blended all together. And then you also will sieve it and paste and take this this biomass, and you also will preserve it for DNA extraction later. So you have then, after this processing, you have two fractions of motile sample and one fraction of the sessile organisms that were scraped from the plates. And you're going to use standard protocol to extract DNA from this fractions using standard kits like the DNeasy Parmax soil kit. And after that, you go through the metabarcoding process. So you amplify a small piece of DNA, usually the CO1 and 16S, just a short sequence, and you do it in a multiplex way in order to reduce costs, so you can do many samples at once, and then you can generate information about all the arms that were deployed. So what we learned using ARMS around the world. So this is one of the first papers that came out using ARMS, published in PLOS ONE, led by Leticia Pleasance and Nancy Noldon and Russi, which was the person that conceived all this ARMS project. So they used information from different places like Heron Island, Lizard Island, French Frigate Shoals, Ningle Reefs, Moorea, and Panama. So they had 93 samples, including 15 arms and 78 coral blocks. So we are comparing the coral blocks, the actual coral blocks, with the arms, only using Crustacea, an example, because they had more information in their database about Crustacea. So in the end, they got over 500 OTUs, or operation taxonomic units, and 40% of these OTUs were single occurrence. So they only appeared once, while 81% were found in a single locality, in more than one unit, but in a single locality. So what we can see is that this fraction of the diversity is very, very endemic to different places. So they are very unique. They are a very unique component of the biodiversity. Another paper that came out, led by Matt Leray and Nancy Noldon, used it and assessed the DNA barcoding and compared the arms deployed in Virginia and Florida, and they left the arms cooked for just 6 months, and they generated a lot of sequences, so almost like 1 million sequences with over 2,000 OTUs. So that's what you get after analyzing these sequences. Using the database back then, what you get is the sequences identified up down to broad levels, taxonomic levels, like phylum or class. So it was hard to really detect species using this approach. And a lot of the sequence, many of the sequences were still undetermined. You could even say to which phylum they belong. So that's a huge thing. So that shows that we know not too much about this fraction of the diversity, which is, as I said, very important. So this other paper shows a comparison of the metabarcoding with morphology-based identification, so we can compare and see if they are comparable, if what you actually see with regular taxonomy correlates with what you get on metabarcoding. So you can see a very good correlation between morphological and molecular diversity. And that's to show that the molecular approach can be used without can be used to speed up the process, because the morphological approach, it's very time consuming. And when you use this molecular approach using DNA sequencing and metabarcoding, you can do it really, really fast. But you still need, during the first phases of the project, to do the proper taxonomy to build up a good DNA database. That's just another example of arms deployed in the Red Sea, showing also that a lot of the components are still unknown, and also showing that the main groups that you can find in the cryptic fauna are arthropods, annelids, and I think mollusks are also appearing a lot in the arms. So I think so arms can be used as well to assess different diversity levels, not only I mean the macroorganisms that are living there, but also other important aspects like the chemical diversity and the microbiomes associated with the reefs. So you can also assess different levels of diversity using the arms to monitor in the long term. So just to conclude what I intend to show, most of the reef biodiversity is cryptic. So a lot of the species there, most are still unknown, so we don't know much about this composition. We have to keep studying them, to keep doing taxonomy of this component. And this component is also responsible for very important functions in the reef ecosystem. ARMS have been a very efficient way to monitor this component. So it's standardized, so it's the same unit everywhere, the same protocol everywhere, so we can compare reefs all across the planet within different depths, different places. So it's a very, very interesting way of monitoring what's happening in the reefs. And of course, you need efforts to inventory and build the database for doing this in a more automated way. And we still have to work on to uncover the functions that are performed by these cryptic organisms, which is still not really understood. So this is our team here in Brazil. There are many ARMS teams around the globe. So this is our team here in the University of São Paulo. So we're working with ARMS in the coast of São Paulo, and we're spreading to start deploying ARMS in the islands as well, in the oceanic islands, and in the more tropical settings of Bahia. So thank you very much. Here you can find my email, my phone number, and my address, and on my lab. I'm also part of the Coral Vivo project. And this project has been funded also by FAPESP, CNPq, and also CAPES. Thank you very much. I don't know if you have a few minutes, we may show them a very short movie showing what the ARMS are actually, how they actually work, and some of the processing. They're just 2 minutes. I don't know if you still have time. We can show you very quickly. Thank you very much.
Thank you, Tito, a lot. Abraham, if you can show us the Tito's movie. Abraham, if you can show the movie, play the movie, it would be nice. Okay. Anyway, okay. Here you go.
All together. ARMS are being deployed all around the world, from the equator If you were going to measure the health of the ocean, how would you do it? Would you count fish? Would you count coral? Would you count crabs? This is an ARMS, or Autonomous Reef Monitoring Structure. An ARMS is a stack of PVC plates separated by spacers that function as prefabricated housing for marine creatures. Think of it as an underwater condo. Their structure is meant to mimic the nooks and crannies found in ocean habitats where the majority of species lives. ARMS are placed on the seafloor for a set period of time. After a year or more, they are retrieved to see who moved in. Researchers take the arms apart, photograph the plates, and run everything through sieves to separate moving creatures into size fractions. Then they scrape the plates into a blender so that the DNA can be analyzed all together. ARMS are being deployed all around the world, from the equator to the poles, and from the intertidal down to 300 meters. Because ARMS are all made the same, you can compare marine communities between places or over time. Along the way, scientists are discovering a hidden world of diversity and even new species. ARMS can also be used to predict how species might adapt to changing ocean conditions like pollution or ocean acidification. We all know it's important to visit the doctor for a checkup. We need to do the same thing for the ocean. By combining simple materials with new technologies, ARMS help us to take the pulse of the planet.
Legal. Very good, very good. Very interesting. Thank you, Tito, for the very nice talk. You know, it's for me, it's very good to see how the technology is improving and helping us to understand the fauna. Thank you a lot. Now I would like to invite the second speaker, Genuario, Professor Genuario Belmonte. Okay, here we go. First of all, thank you for being here, Professor Genuario. Professor Genuario has a degree in biological science and natural science, and he works at the Department of Biological and Environmental Sciences and Technologies from University of Salento, Italy. Professor Genuario works with zoology and ecology of plankton species, and also with marine debris and coastal management. So thank you again, Genuario, to be here, and you can start your talk. You have to unmute. Yes.
But. Sorry. Oh, yeah. Now you hear me?
Yes, now we hear you. Thank you.
Sorry for the misunderstanding. Okay.
No worries.
I have to start with the presentation. Here it is. And just to connect with the preceding presentation, I would like to extend this approach to the cryptic biodiversity, because I work from 30 years on the hidden biodiversity, because when these species are not playing a role in the ecosystem, they stay elsewhere. they stay elsewhere, even in the same place, because probably, or many of them, they simply are resting. They simply are passing a time more or less long to waiting for return in the play. The problem is that of the plankton, because I am a planktonologist. So the plankton, we are habituated to speak about the plankton as a singular entity, only one entity. So the zooplankton is there, and he is obsolete, and it oscillates in time. So abundant in some season and not abundant in some other seasons. But the zooplankton is, or the plankton in general, it is composed by many single species. And the plankton is there, but species can be absent time by time from the same field in the nature. So marine plankton ecologists do not care for the destiny of plankton components, the species, when they were not there. This is a story of my studies. So this is an approach already overpassed today. But in the past, 30 years ago, the plankton ecologists didn't care for from where the species arrive. And even modern textbooks base some aspects of marine plankton ecology on this mystery. I studied in a time when the phytoplankton was produced by nutrients. So, and you understand, if you are a biologist, that is not possible for a living organism to be produced by a non-living material. So, but this was what they told us in the past, from many nutrients, many phytoplankton. And, but where is the phytoplankton when the nutrients are not there, or when the plankton is not there? At the present, we know that diatoms, dinoflagellates, ciliates, rotifers, cladoceans, copepods in temperate coastal seas overcome the adverse season as resting stages. We call these resting forms cysts for the unicellular organisms, the protoctista, which and cysts themselves, generally after the sexual reproduction. Or we use the term resting egg for metazoa embryos, which in turn could be considered encysted inside the egg envelope. How many morphotypes of resting stage? So this is another aspect of the biodiversity. The diversity, the morphological diversity. Sorry, I lose my presentation. Are you there? Yeah. So how many morphotypes? You can see from this simple approach that morphotypes, even of these cysts, is very variable. Resting stages differ from the corresponding active stages, and their morphology is unexpectedly diverse. Sediments in confined coastal areas are marine seed banks like those of terrestrial forests. So we can search plankton in the mud, in the sediment, because there we can find more plankton than in the water column. This is an aspect at scanning electron microscope of the morphologies, but we can find some rules. For example, resting stages have typically sculptured, so they show sculptured coverings. On about more than 200 described resting stages, we found 160, so the 80% showed a sculptured covering. And the sculpture can accelerate the sinking among diatoms. For example, spines allow the clinging of many individuals, and so they favorish the sinking. Or the sculpture allow the passive transport, when spines allow the clinging to floating objects or migrating bigger organisms. The spiny surface is a constraint typical of resting stage and common in different realms. A constraint. Why? Because in a normal egg, this is a copepod, for example, in a normal subcutaneous egg, the surface is smooth or just a little bit pointed. But in resting eggs, the surface presents these trees, this spine, very complex. So when the species, when the same mother produces a resting egg, she performs another genetic program to realize a different egg from the point of view of the chorion morphology. The sculptures. Why the sculptures? Because they extend the contact surface when the spines are hollow and contain cellular material. The sculptures avoid the scraping. So when the spines represent a device of passive defense, the cysts buried under 30 centimeters of sediments are still viable. And the sculptures avoid the predation, probably also against chewers and or drillers. We don't know. This is an aspect that we don't know. And the sculptures allow the survival of the first embryonic stages because the rest is entered in an advanced embryo stage. If the sculptures retard in some way the sinking, they help the realization of the first early stages of the embryo before to enter in the rest when he will reach the bottom. This is just an example of where are these resting stages. This is Italy, probably you recognize. And if you see this table aside, we find many cysts where in the site F, E, and C. Because in these sites, we find more than 50, 48, or 26,000 cysts per square meter. But attention, this is in the first centimeter of sediment. If we observe the fifth centimeter in the sediment, or the 10th centimeter in the sediment, we find that this percentage, this abundance, doesn't diminish. You see in C, and even it can be double as in the case of A, because the first centimeter is interested by the exchanges, so germination and reformation of cysts with the water column. But the 5th centimeter and the 10th centimeter below the sediment surface, they stay there. So they are not interested in this subtraction. And we find even more cysts in the deep sediment. Where are these situations? Mostly in confined environments, so harbors, lagoons, and inland seas, because there we obtain this accumulation of cysts, of seeds, of animal seeds in the sediments. Even along the vertical profile, we can recognize that some species, this case is that of Lingulodinium polyedrum, is a dinoflagellate. You can see that we have an abundance at 3 centimeters from the sediment surface. But in the case of this Acarcia, is a copepod, a crustacean, we can find an abundance at the 7th centimeter below the sea surface. So each population, each species has a different story along the sediment vertical and along the period that this vertical of sediment represents. Their abundance varies apparently without a common pattern along the sediment column. Again, if we consider the many different devices to collect plankton together, for example, in this study of the South Adriatic Sea, we found 26 taxa exclusive of the water column, but 47 exclusive of the sediments as cysts, obviously. and only 16 in common between water column and sediments. Again, other 78 taxa were not in common with sediments, and other 18 taxa were not in common with the water column. So you can perceive that 47 and 18 are a component of the biodiversity subtracted to the analysis of the water column. If I collect plankton, I cannot perceive the biodiversity, the planktonic biodiversity, which is stored inside the sediment. This is the story of that cruise. For example, in 14 different stations, we found that only cysts in the sediment, and only in 2 cases also the presence of active stages in the water column. This is the case of Scripsiella trochoidea at dinoflagellate. And more interestingly, in Labea strobila, a ciliate, we found the presence of active stages all over the basin, but not in the station 10, where we didn't find the active stages in the water column, but we found plentiful of cysts in the bottom. Incredibly and simply didactic, this kind of example, because when the species is not in the water column, is in the sediment, and vice versa. An age of 40 years has been attributed by Nancy Ellen Marcus about 25 years ago to a calnoid eggs coming from an anoxic basin of the northeast of the United States coast. And an egg of freshwater calanoid, this is a crustacean buried in a sediment layer of 300 years old, hatched in lab of Nelson Hairston. This is a species of freshwater, okay. But you can start to understand the importance of this phenomenon. The species, the organisms can wake up even after centuries, not only the adverse season. This is an experiment that we had in the past in our lab, and we obtained this cyst in A. In B is just the covering, and in B2, the moving organism with the flagellum not visible here. And a dinoflagellate, yes, but this is a cyst that hatched from a layer of about 100 years ago. So even in our lab, we observed this possibility for the organisms. I can tell you stories about the immunobiology. So because there is a resting form, they are organic material, viable organic material, but they are not living. So they are viable, but they are not living. So they can, how they defend themselves. For example, with this immunity, but the immunity is a living process. So this is a passive immunity. And this is a story that we studied in resting stage in cysts of Artemia salina, for example. And we found lysozyme-like and trypsin-like molecules in the shell of these eggs, in the shell of Artemia cysts, which do show it to have not only a mechanical role against environmental aggressors, but also a chemical defense. Even if we use disinfecting procedures, we are not able to kill the embryo inside the cyst because the cysts are very resistant. So what this means for a plankton ecologist, the disappeared population can return to be present in the same environment even after long periods of absence, because and if they have resting stages in the environment but elsewhere, so in the sediment, not in the water column. Plankton communities of confined environments have a resilience higher than the previously suspected. embryos existed centuries ago, when allowed to complete their development and to form an active stage, can recombine their DNA with present-day individuals. This is a movie story. This is a movie from the science, for the fantastic science, but it's the reality in the plankton. The long time rest allows organisms to pass over adverse conditions also in the space and to maintain gene flow also among distant populations. According to some researchers, as Fenchel, Wilkinson, and Finlay, the small-sized organisms tend to be cosmopolitan, so the biogeography does not rely upon them. Why? Because small-sized organisms probably have cysts, and with cysts, they can pass through each ecological barrier. Why geographic distribution and dispersal ability are due to resting stages? Probably. Here I can recognize a copepod, 2 dinoflagellates, and a ciliate. It's incredible. But we are able to recognize species and taxa from the morphology of resting stages. Cysts are produced to avoid the habitat hostility. So they are pre-adapted to go beyond geographic barriers or to ignore stress conditions. They are insensitive to vectors environment, so they can stay even inside a vector if the vector is an organism, so the gut, or even inside tanks if the vector is a ship. And as a consequence, the geographic distribution of species could be affected by cysts and or rest features. In studies of Marcos and of Ichime Montani from Japan, it has been found that polychaetes and fishes do not digest the ingested cysts. And when the cysts appear in the fecal pellets, they regerminate. The passage in a polychaete gut enhances the hatch rate of some dinoflagellate cysts, again, in a study of crank. So the cysts need to be ingested, to be activated when they stay, again, outside. In Eurythemur erdmanni is a copepod, mature females offer themselves to the predator Menidia is a fish. The fish eat on females. Females, they have an egg sac with resting eggs, and these resting eggs, they need to pass the gut of the fish to be activated. Incredible. So the dispersal is affected by mobility of predators. Birds and wind are not the only vectors in the marine environment because we have also the ships, so the ballast waters, a huge problem in the ecology of the oceans of the last period we discovered, because this is a study of more than 25 years ago, about 4,000 non-indigenous species were recognized as vehiculated by tank ships all over the world oceans. Species are transported mainly as resting stages, obviously. The active form cannot support the harsh condition even of a tank in an oceanographic shape. So the solution is the resting stage. And something about the ecology of coastal areas. This is the profile, the vertical profile of a confined area and of the adjacent open sea. You can imagine these gray circles as the plankton. So more plankton inside a lagoon, small quantities of plankton outside in the open sea, but the plankton is there. The species is the same because we have exchanges between the sea and lagoon. But when arrives a problem here, for example, an environmental problem, the species disappear from the lagoon because the lagoon is exposed to problems more than the open sea. The species is preserved in the open sea, and what happens? After the restoring of favorable conditions, the species recolonize the lagoon. This is the ecology of our past. But today, I can assure you that I'm convinced that it's not this is the story. The story is another, because during and before the harsh period, the zooplankton, the plankton in the lagoon, they produce cysts, and the cysts go to the sediment. When the problem arrives, the plankton is already disappeared because the plankton produces the cysts. The plankton is able to produce cysts in a predictable way because the organisms, they perceive the astronomical messages. So they know when the winter is arriving, they know when the summer is arriving. And before the arrival of the harsh conditions or of the opposite season, they produce cysts before the arrival of the problem. When the problem is passed, from the cyst, we have the regermination and the reproduction of the population in the lagoon. And due to the huge abundance of plankton inside the lagoon, we have an exportation toward the open sea. Is exactly the contrary of the ecology that of my generation we studied. So from the sea, regeneration of the lagoon. No, we have an injection of biodiversity and of organic material from the lagoon to the sea. The same we can use for a solution, or to propose a solution even for the oceanic plankton if we stay in the poles. So if we recognize the ice cap and the ice, the floating ice, it contains such void spaces, and these are the sympagic space. Inside the sympagic space, we have to imagine the content of cysts. When the ice melts, we have the freedom for the phytoplankton. So the phytoplankton is not there. Please pay attention and remember that in the polar seas, we have the night, a night for months. So it's not possible for the phytoplankton to stay without the sunlight and to reproduce and to survive. So the phytoplankton is not there. The phytoplankton disappeared. Where is the phytoplankton? Cysts is the explanation. And if we have cysts, when the ice melts, the ice makes free the cysts, and the cysts germinate the plankton. So during melting and during the ice formation, we have the encapsulation of encysted plankton in the sympagic space. So the cysts can explain the mystery of the appearance of phytoplankton in the North Pole, for example, where we have a pack ice and a very deep water column. So even if the cysts go down in the bottom, they cannot perceive the stimulus to go back in the water column because they don't perceive the light at 1,000 meters below the sea level or at 2,000 meters below the sea level. They cannot perform information from the environment. They can perform information from the environment if they remain in the ice. What is the message, the final message? You see, this is a desert. When it rains, and it rained here just few hours before or the day before, all the desert is flourished because even in the desert we have seeds. And they stay there and waiting for the arrival of favorable conditions, a small quantity of water in this case. What we have to do to prepare an atlas on what we are doing, what we are working at, to prepare an atlas of unknown resting stages from the neritic plankton, about 500 recognized at present. We have about 500 resting stages to recognize at present in the dimensional range of 3,300 micrometers. We have to understand better the physiology, the immunobiology, the biochemistry of diapause in different taxa to acquire information about the import, export of cysts in the marine seed banks, because we have to create this new ecology. The resurrection ecology is the ecology of the marine seed banks, not the ecology of plankton, but the ecology of the resting stages. So how many they arrive and how much they hatch from the sediment. To find the direct consumers of cysts, we still are searching for a direct consumer of cysts who uses this organic material in the sea. And could, for example, a nematode of the Meiobenthos be responsible of the plankton dynamics if the nematode eats specifically some cysts and some other not? We have to describe also the total biodiversity of sites and habitats, also if we consider its temporarily unexpressed fraction. This is the temporarily unexpressed fraction of the biodiversity. And we will be able, if we will study this particular aspect of the life cycle, to rebuild the story of plankton communities in basins where cyst mostly accumulates. For example, the Black Sea. Cyst accumulates there, and we simply reading a core of sediment, we can rebuild the story of the plankton in that basin. This is all. I thank you for the attention. And I'm ready for question, obviously.
Okay, thank you, Genuario. Very good.
Are you there?
Yes, I'm here. Thank you for your nice presentation. It's very good to see that we cannot understand plankton without looking at the cysts. We have questions after the final talk. Okay, so thank you again. And now we're gonna have the third and last talk from Rafael, Professor Rafael Sardà. Just a second. Yes, Rafael is there. Okay. Professor Rafael Sardà works at the Center for Advanced Studies of Blames in Spain. Okay, he's a biologist with a PhD in marine ecology. He has almost 30 years of experience teaching environmental management courses. His research deals with the coastal zone management and ocean governance. And he works in the frontier between the social and ecology. So thank you, Rafael, for being here. And you can start your talk.
Hello. I think I'm gonna try to share my screen from here. Can I try? Because I just did something that maybe it works. It would be much nicer if I can manage my presentation. Can I try?
Yes, we can try. Let's go.
Okay. Did you see it now?
Just a second.
No. You did not see it?
No, I can't see that.
Okay. So then it's a pity because there is a lot of interactivity in this presentation and we will miss that. But anyway, sorry about that. Yesterday it was working and now it is not working. So it's amazing how so how things are going these days with technology. Well, anyway, nice to be here with you. Thank you for inviting me to this particular talk. It would be much nicer to be in Fortaleza. I visited Fortaleza many years ago. I would like to be there instead to be here in my center doing this online, which is so boring sometimes, and you cannot see the faces of people that is in front of me. Anyway, thank you for the invitation. My talk is going to be really different from the previous two, because I'm going to jump from the ecology of living organisms or the ecology of resurrection into something which probably is much boring because it's just pure management, but I tell you as is important. So we will be going from knowledge to the way in which this knowledge should be transferred into real management for conservation. So you can just put the presentation. It's a pity I say it, because there was some interactivity in this presentation, and I'm gonna miss this just by saying, okay, pass. We'll be just doing pass.
Sorry.
But anyway, so my talk is, if you can just introduce the presentation, it's about the application of a strategy that the United Nations launched at the end of last century in relation with the management of all these issues that are together between the social societies and nature, and the way in which this strategy is applied to management. So I will start with the second slide, and now you will see why the interactivity. In this year, in 2020, they show up two papers that capture my attention and that can express really the dilemma that we are running these days in the world. On one side, probably you know that in Nature, there was a paper this year by Carlos Duarte and collaborators about rebuilding marine life, the way in which we can rewild the world, the which in way, if we are able to alleviate the pressures that we humans are sending to the ocean is going to restore quickly, rapidly, and probably in a very good way. On the other side, you can pass next. On the other side, there was another paper by Humphrey and the collaborators in the Stockholm Resilience Center that were developing the needs and the demands of people in the ocean for food, for materials, and for space. And this paper, what it's just saying is that basically there is a high demand and is going to increase this demand, and they call it the blue acceleration for the demand of people in the ocean. So you see, we live in a world, and you can go to the next. We live in a world which has been defined and they enter in the Anthropocene, in which there is this dilemma between conservation and activities, and the management of these activities and the management of the conservation. In a recent book that I published with a colleague, an Italian colleague last year, we were just dealing about how business should change the way in which they do things, change the way in which they do sustainable corporate sustainability, to try to address this particular dilemma. And to tell you, we live in a world in which people is increasing 1.2% as an average the last 18 years. We live in a world in which the economy is bumping at least 3% per year. As a consequence of these two things, we live in a world that everything is accelerating. And as you know, because we are right now in a very good example, all this acceleration is in addition connected by many forms. So we live in a world that is connected. We live in a world in which also scientists are advised people that we have limits, these planetary boundaries that has been with us for decades now. And then we live in a world in which we lost resilience. We have lost the resilience of the social ecological systems, and we have plenty of problems. In June, we go to the next slide. In June, we presented with a bunch of people from an Italian foundation, just from the One Ocean Foundation, a paper or a work we did in relation with global business and the oceans, and the way in which people benefit from the oceans. You see here a lot of data, a lot of statistics about many things that the ocean is giving us, much more than in economic terms, much more in employment that probably you should think, because there are those not just direct employment, this is indirect employment and benefits. You can go further. And then we presented this in United Nations, and the idea, you can go next, please, next. And the idea was that even in business, even in the activities that we have in the ocean, there are right now, of course, we have pirates, we have people doing business as usual, we have people that we will like that they were not there, but in general, there is a sense that it's time to do something different. And we launched this idea of business for ocean sustainability. Now it's a pity because this map goes with a video, and you will not see the video, but the video, this map is coming from the paper of Carlos Duarte, expressing the way in which we have been conserving areas in the ocean. Although one of the goals of the Sustainable Development Goals, the Sustainable Development Goal 14, says that by now we should have conserved 10% of the marine ocean, at least in water jurisdictional waters. Still, we have just 6% conserved. We didn't do our target. And scientists, as you know, we are asking for a 30% conservation in the years to come. It's true that we are accelerating the way in which we are conserving areas in the marine domain, and the map was ready to show this indication, but still we are far from accomplish what scientists say that we should do. In addition to this protection, for example, there are places in the world like the European Union in which we have the idea to keep with a good environmental status. United Nations is right now presenting this idea to have a sanctuary in the high seas. And if you go further with this slide, you pass next and next, well, anyway, there was exactly here, which is also a phrase that we had in the book that I told to you about the need of people, the need of societies, the need of going into a sustainable development path in which definitely we work with the idea that we need to maintain the integrity of these natural systems, to keep the potential of these natural systems to give us ecosystem goods and services. And if we go to next, this is basically the idea of this strategy. Go to the next one, please. This is basically the idea of this strategy that I want to bring right now to you, and that was launched by United Nations in 1998, at the end of the last century. I always start my talks with this timeline. This is a kind of timeline that shows to us in which way the environmental thinking of people have changed over years during the last 100 years in the world, by going from a social paradigm to the actual present social ecological paradigm that stress the importance of dependence, not just about our impact on nature, but the dependence that we humans have from nature, which was extracted in the Millennium Ecosystem Assessment. The ecosystem approach was ready to work with this idea of this dependence. We live in a world for which we are dependent. So if the earth cannot serve us, we will not be just living well. So United Nations sent the message, and this message has been with us for 20 years, but still the application of this strategy, this integrated strategy of the relation between people and nature, is not is a little difficult to understand, or at least has no a real application. The idea of the strategy was that was able to go into managerial aspects, that was able to enter in management applications. So you go to the next one. This strategy is based in what is called the Malawi principles. We are not going to have time right now to go through all these principles, but it's very easy to find these principles in whatever, if you Google a little bit. But those are the strategic principles in which we should base our relation with nature. And starting to think that from the idea that we are part of nature, we are not something isolated from nature. We are in nature and we are served by nature. So that's why we are dependent. And the entire concept that is included in this strategy is this concept of ecosystem goods and services. So if you go to the next one, I will show you on a slide in which basically developing aspects. Just pass to the next one. The next slide, please. And you can just go one more and one more, just to show all the pictures that are here. Yeah, that's fine. And sorry about this interactivity that we lost from not having the possibility to present from here. But anyway, this slide is showing to you that if you go to policy, this is European policy, the conservation policy, the maritime policy that we have here in Europe, or here in the Mediterranean, because the Mediterranean Action Plan is a strategy for all countries in the Mediterranean, independently that are in Africa or in the Middle East or in Europe. All this policy that has been launched in the last 20 years has the ecosystem approach in the preamble. All the policy, conservational, environmental, maritime, all these policies says that at the earth of the development of the policy should be the ecosystem approach. So we have a need that to include this strategy into real applications. So if you go to the next slide, what we have here is a kind of explanation. Yeah, you can put two more, please, because exactly, that's fine. What you have in this slide, it's a kind of explanation about why, if you have this just clearly stated in policy, the application is very, very weak. So there's not proper governance framework, there is a lack of common language of application, there are sometimes the applications are very, very independent one through the other, so they are conflicting in the way they apply many things, because they apply things differently, sometimes with confusing visions, response to a lot of different fragmented policies that are in conflict with this one. Languages are sometimes very different. If you go from the social issue or from the natural issue, sometimes you find this Babel Tower paradigm. And there is no proper visions developed for management, which at the end makes that the application of the ecosystem approach is highly subjective and depends basically of the manager that thinks that can apply a particular issue in place. So we end up with this idea that we should try to reconcile the theory with the practice. In Europe, I will introduce this in few slides. In Europe, we have been working with a big project called No Cysts, started around 10 years ago. It ended around 6 years ago. And then what I'm going to present to you is basically a tool that we developed for the managerial application of the ecosystem approach into the marine domain. And I will bring this into conservation issues in the marine domain. And for the last 5 years, I have been just making a lot of conference. Right now, I'm going to just talk about marine conservation. No, please, the previous one. Now I'll go to the marine conservation, but the tool can be applied to other things. But in different regions and with different managers, about the idea to bring a standard tool in order to apply this ecosystem approach that could be used globally, and everyone could be working with the same tool. And concerning with MPAs, marine protected areas, or network of marine protected areas, I was related with this NEAMPAN, the one in Asia, and in the region of Asia Pacific, or the one that we have in the Mediterranean. And when you talk with these guys, all of them, they require much fundamental management tools to apply these concepts. So if you go to the next one, this is just something that we did in the past. This is what right now developing in Europe, in the Mediterranean region. We are preparing this MPA forum process that, depending on the COVID, will be placed next year or in 2 years. And we develop a kind of roadmap in order to develop in a much more agile way the way we manage marine protected areas in the Mediterranean, and with some kind of questionnaires and platforms, we develop these limited factors, from which many of them takes in consideration the issue about real management. So if we move into the next one, I will just say that, just pass to the next one. The issue about, well, you can put all of them and then I will explain. The issue about the ecosystem approach and the application for management, which basically is called ecosystem-based management, it's an issue per se, because when you go and talk about managers, many of them did not differ between the ecosystem approach and the ecosystem-based management. And it's a pity, because one thing is strategy and principles for this strategy, and the other thing is the management of the strategy, which is a tool. And there is a confusion with the idea that the strategy per se is the tool. So if you don't have a tool to apply the strategy, you will end up in a very subjective way to put this in practice. So we thought about that years ago, and we tried to develop something that could be useful to develop a standard tool for application of the strategy that, as you will see, we call it the ecosystem-based management system. And basically, what we wanted to do is a managerial tool. You will see this tool right now, at least the idea of what it is. And the idea was that in this tool, by using this tool, you could, with this tool, you could rely all things that were, and now we will see application for conservation of the marine domain. You will see all the principles of the ecosystem approach internalized in the tool. So if we jump into the next one, I will introduce this tool, which is called, the next one, please. It was called the ecosystem-based management system. And the idea was simple. The idea was that we wanted to have a standard tool, something very similar to what it is up there for business, for organization, in the terrestrial domain that is used, the ISO rules, something similar to that, but that could be applicable to the introduction of the principles of the ecosystem approach. So we end up with an adaptive management tool that runs under the quality and risk management schemes that normalize, standardize a common set of instruments, with basically has a common language that if everyone is using, everyone can understand what we are doing, and that is intended for real practice. So that's the paper in which we describe the system. And going into, for example, into integrated coastal zone management, going into marine conservation, going to beach management, you can apply the tool. So the tool is the same, but the adaptation of the tool to the social ecosystem, social ecological system under management is what it changed this thing. So what is this tool? If you go next one, this tool works with 3 pillars. There is, and the idea of to have these 3 pillars is because you need to have a structure in which all principles of the ecosystem approach can be easily observed. And these 3 pillars are a managerial pillar, an information pillar, and a participatory pillar. You go to the next one, and you will see the managerial pillar. And this managerial pillar, it works. It's basically the engine of the tool, and it works with procedures, formal procedures, standard procedures. I don't know if you are familiar with an ISO 40001, which is one of the most easy and adaptable tools that we have for environmental management of whatever organization. So something similar to that. But what we decided to do is to change a little bit the clauses of the system of the ISO 14001 for others that are working with the concept of ecosystem services and that are working with risk quality systems. So it's a combination of environmental management quality and risk systems all together. So that was basically the managerial pillar, and a standard set of planning, doing, checking, and reviewing things, which is a normal management plan. And this managerial pillar was accompanied by 2 other pillars, an informative pillar, which is needed because if you need to have decision-making systems, you will need information in order to base this decision-making process. So an information pillar that for the purpose of marine conservation obviously needs to go with an spatial data infrastructure and a platform of indicators that responds to an accounting framework, which is we follow this driver, pressure, state, welfare response as the basic elements of this information pillar. And then a participatory pillar, which is the 3rd one, in which we just include many aspects that are related with the principles of the ecosystem approach, about participation, about governance, about the possibility to develop a consensus in the vision to which we want to address. So that was also the training systems that you are needed and the educational systems that you are needed to run the system. Okay, so as a key aspect, it's a pity that we cannot pass the video because you will ask to me why you put a tiger in a slide. We are talking about the marine domain. But in one of my visits to Korea for a meeting of Niampan there, the guy, the director of the Shikote Alin, is a pity of the video because it's a video. No, you cannot pass, but the director of this park came, and of course, this is a Russian terrestrial marine park that is near Vladivostok, very amazing one, and very big, where the basic animals that are living there, we are talking about something that is very north, is tigers, and those are forest tigers. And besides the fact that they can predate on deers, I saw images, fantastic images of these animals predating on seals in the rocky shores, which, by the way, that's amazing. I never saw this before. So talking about marine conservation, sometimes you end up with situations in which this guy can predate on marine animals too. Well, anyway, that's one of the key aspects of this EBMS is to develop this vision driver process. So we want to reach a place in our management issue, and this place that we want to reach is not the one that we have. So we need to be able to develop through this managerial process a kind of adaptive plan in order to get into our vision for the future and to stay there with this vision. Another key aspect of the EBMS, besides the vision in aspect, which is basically the first principle of the ecosystem approach, is the work with ecosystem services, and sometimes even with the issues related to the socioeconomy that is behind the benefits that we get from nature. So the issue to work with ecosystem services as the main point in the managerial pillar is important. And of course, all these things that you can read here that are related with the way in which we understand, we know about what is an ecosystem and how this ecosystem are benefiting us. In relation with the managerial pillar, the key aspect is that we are just dealing with ecosystem services, keeping this ecosystem service, analyzing the risk that these ecosystem services cannot properly make their functions. So what we are basically delivering is regarding every marine conservation area or the network of the areas in which we are working, what we are trying to set up is what the risks are involved because of many things that can happen in this place that impede us to reach this vision that we wanted to have. This is the most important part in the managerial pillar, the relation of risk management and ecosystem services. And we construct a tool that was called DEMA as decision making. In the next one, in the information pillar, what we work is basically with GIS, of course, for any area, and then we work with this accounting framework, the driver pressure state welfare response, in which we can relate all the issues about what is very, very important in the area, which is the state of the area in relation with the pressures and the benefits we get from this state, and analyzing all the hazards and human activities that can influence this particular issue. And from the participatory pillar, which is the next one, a lot of tools that basically brings formal consultation processes that makes people more engaged on these conservational areas, and collaborative approaches in the fact that one of the problems we have with marine protected areas is lack of resources. So because those areas are delivering a lot of ecosystem services that benefits to us, someone needs to pay from that. So the payment for this ecosystem service and the collaborative scheme in order to make the areas functional are important. So these are the most important things or topics in the participatory pillar of the tool. And just to conclude, in the next slide, I want to make some clear remarks about the good things that you may have if you introduce a standard tool in the application of all these aspects. We should consider that the ecosystem approach is a priority of United Nations. Was delivered like the way in which people and nature should be linked in policy. So the ecosystem approach has entered in many policy tools worldwide. So is there to be used, and this strategy needs to be brought into areas if we are talking about conservation, and into the management of these areas. And these areas can be working with this tool isolated from the rest, but also inside the networks of areas in which normally they are, or in a global network of networks that we have. So the main things that this tool, the main things that needs to deliver thing here is this is a standard system. So if you work with this standard system, you adapt the standard to your particular area or to particular network. But you have some things that you need to do just following these procedures. It's common language, it's common tools, it's common. There is this issue of commonality that will allow us to avoid this Babel Tower problem that suddenly we found when we talk in many conferences and congresses between people that are theoretically using the same strategy. And finally, it's scalable. So you can use, as I say, you can use the tool for a particular MPA, but you can use the tool for the network in which this particular MPA is there, or you can use this tool at a global scale. And if you use the same tool applying to your proper work, if you use the same tool, it will be very easy to you to talk with the other guys because everyone share the same language. So that was the thing that I wanted to just say to you. A really different talk with the two previous ones, and basically in which way conservational issues can go into management. Thank you very much for your patience, and thank you very much for the invitation again. And now I'm waiting for your comments and questions.
Okay, thank you, Rafael. Very nice talk. I think your talk complements the Tito's and Genuario's talk, because in some way we need to use the information we get to the ecosystem. And you present a very nice and interesting tool to use this kind of information. Okay, so everybody's here now, and we have lots of questions. So let me see. Okay, I will start. I have all the questions here. Okay, the first question is for Tito. Guys, if you have like comments, you can comment after the other talking. Okay, but the first question is for Tito, and it's from Tatiana Garcia, and she's asking, do you think that the molecular approach could be used in ecological monitoring? As you said, morphological analysis take too much time.
Yeah, sure. It's been used actually in many different contexts. You can use molecular tools now for monitoring like stream organisms, sediment, or anything. The only thing that you must have in mind that you rely on a good database. So yeah, you can use the molecular tool, but you still need the taxonomic work to feed all the database with all the proper information so that you can establish your monitoring using molecular tools. So it can be applied in many, and it has been applied in many different contexts all around in different ecosystems, terrestrial and marine. And it's a powerful tool, but it doesn't work by itself. It needs information from other areas as well. But yeah, you can do that.
Okay, thank you. Any comments, Genuario, Rafael? Okay, good. The second question is for Professor Genuario. What I'm doing, I'm pasting the questions at the chat. We have like a private chat, and you can see the questions there. So the second question is, Genuario, how much we know about the role of biogenic formation as rhodolith beds, seaweed beds, seagrass beds, regarding this function as sea banks in comparison with no vegetated soft bottom?
I don't know if this is the same problem, because the rhodolite beds and the seagrass and so on, they can catch resting stages, because inside a prairie of Posidonia beds, for example, we have the smoothing of the hydrodynamism. So there you can accumulate cysts, but they have not per se a role as resting stages, the rhodolite beds, for example, the seaweeds and so on. Okay, so they can play a role in preserving, in the storage of resting stages. Yes, we have to use the approach of Titus, for example, just to find a way on how to collect such a teeny organism inside very complex structures. Is the same problem? Yeah.
Yeah. I was thinking about that. If you can take like the sediments and do like this soup of organism and do like the metagenic analysis, but you have a problem.
Solution.
Yeah, but the problem is you need like the taxonomic analysis as well, and I think that's the hardest part in your side. Don't.
Can I tell something about the use of metabarcoding and so on?
Yes, you can.
Because the metabarcoding is a powerful tool to perceive the biodiversity. Yes, okay. But how a committant asks us in this moment, please tell me how much is the biodiversity in this site. So we are asking to the metabarcoding method, please tell us how much is. So the problem in this is not a problem of information. The information is there, but we lose the experts. So the guy who studied the nature, they are expert of biodiversity. If we use a tool, we lose the expert. This is a problem for us, not for the commitment.
Yes, very, very good. We had a question actually regarding that from Sergio Rossi. Sergio Rossi asked Tito, Tito, do you see a contrast or conflict between the, let's say, formal morphological taxonomy and metagenics? So Genuario did some comments about that. And
yeah, well, exactly. There is no actual conflict, because you rely on information that comes from morphology. There are different. Actually, the molecular work is a tool that you can use even for taxonomy, using also morphology and other aspects of the biology of an organism. We still have to keep your focus on the biology. If the tool is getting more attention or is getting more focus than the biological aspect of the problem that you're investigating, then you're going the wrong way. So as long as you are applying the molecular work to investigate and as a tool properly applied to investigate a problem in biology, it's fine. There's no conflict. As I say, at least for metabarcoding, you still need the taxonomy to feed the database. And after you have all this processed and so on, and you have all the information, you have to interpret all this information in biological terms as well. So, yeah, you cannot, just think of molecular per se as a tool. But we need to incorporate bioinformatics in our work even more. So this is also a challenge, but not alone, not by itself.
Beautiful.
If I can say something.
Yes, of course.
Yeah, because I really think that there is at least one conflict that is not in the science part, but is in the evolving of the science part. I'm 63 years old, right? I have been. I started to work around 40 years ago, and I did my thesis in polychaetes. So right now I'm a sociologist working in application and in management issues. But at the very beginning, I did my thesis in polychaetes. And because of that, this is my hobby. I'm not dedicated my professional career to polychaetes now, but it's my hobby. And in polychaete science, I still have a name, and I go to the to this international conference of polychaetologists. That we are in the 13th conference, and there are every 3 years. So I have been there for 39 years. The conflict is that every time I go to one of these conferences, we are less people. The problem with the genetics these days, with metabarcoding and things like that, is that they are not opening professional careers for the traditional ways. So I don't know in Brazil or in South America, but in Europe or in the States, you cannot teach a guy to be systematic because you just are putting here in front of something that will not allow to him to have a position. So we are losing the traditional guys, and this is an important issue because in few decades we may end up with no people able to morphologically classify animals. And this is an issue. This is something an issue because you can make much more papers if you go into doing this kind of analysis that if you go into the traditional classification of species. And from the management of science, this should be an issue for the big managers.
Very good. Very good comments, Rafa.
Yeah,
very nice.
Agreed. I agree totally.
Yes. Okay. Let's move for the next question. Rafa, that's from Sergio Rossi for you. Okay. Rafa, in developing an upscaling plan, example restoration, do you think it's important to consider local people, awareness, and participatory tools? How we have to do that?
Sorry, I couldn't understand a little.
Okay. You can read the question in the chat.
Oh, okay.
We have a little bit of noise in the background.
Okay.
We just look to your.
This is very important because many times people wants to people in general, society wants to know why we are protecting the environment. Maybe they know very well, but why we keep the environments that are not possible to visit, for example, with what we are delivering sanctuaries, why they normal people cannot go into a marine park to enjoy nature because you need to protect, why you cannot pass to so awareness and education is something that is not shouldn't be just reserved for schools and for teaching and for education at this level. Awareness and education needs to be also included in marine conservation. And when we keep areas, protected areas in the marine domain, we need to show clearly why we are doing things and show clearly in which places you cannot be active doing your recreational aspects, and you need to explain that why. And of course, in order to make awareness of people, to teach people, to educate people, you need resources. So someone needs to pay for that. And many people does not understand why they need to pay for this issue particularly, and why some public funds needs to be dedicated to that. So, of course, in whatever conservational plan, you need to bring some tools that allow people to know what these areas need to be kept or restore, and it is really important, because if people is not educated on that, will not follow the rules, and at the end we will not be able to have these areas, because we cannot put the army in front of the area. So the people need to let me talk about one thought that came to my mind, exactly the same that the tiger, in the same area, because the talk of this guy impressed to me a lot. This guy went into Korea. He was a Russian guy. He went into Korea to show a wonderful presentation about whatever animal you can find in this northern hemisphere, big ones, fantastic tigers, bears, deers, whatever animal, seals, orcas, whatever animal you can think in this place. And the main worry that this guy had in the conference was that they needed weapons. They needed weapons to protect the space from people, because there was a lot of people with illegal hunting, and a lot of people entering to fish in the ocean, and they wanted just to have more weapons in order to protect the space. So we are talking about marine protection, or about nature protection, and you end up talking about these issues. And this is just a problem of education and awareness of people about this area. So, of course, this is important, more than we thought, probably.
Very good, Rafa. Okay, guys, any comments? Okay, so we can move to the next question. Is from Tatiana for Professor Genuario. What do you think about the estuarine environments that are also a confined area? The cysts also could be important to recall after drought events like occurrence in Brazilian semi-arid coasts.
Absolutely. Absolutely, yes. Because the resting stages allow, for example, in freshwater ecosystems, allow the replenishment of fauna of before dried lakes. So when a temporary lake replenishes a gain of water, the plankton reappears. So why? Because cysts stay there, not in the mud, but in the soil when the water is not there. So the powerful possibility of resting stages to repropose the life is incredible. Somebody also hypothesized that the life restarted after the last extinction 65 million years ago, based on resting stages, because only resting stages can allow an organism to stay there and to wait for the return of favorable conditions after 5 years, 10 years, 100 years. They stay there and wait for the restoration of suitable conditions. The resting stage is a tool to explain many phenomena on the persistence of life on the Earth, on the planet.
Okay, very good answer. Any comments, Rafa, Tito? No. Okay. So next question is for Tito. It's from Jadson. Tito, as rhodolite beds are a transition from soft bottom to hard bottom, can you use arms to analyze its cryptic fauna, or it's hard to anchor in arms on these environments?
Yeah, well, we can use, it has been used in different bottoms. But one must bear in mind that although we want to imitate the crevices and the reef environment in the arms, they do not replicate the conditions inside the reef matrix or even within the rhodolith. So the first step would be to check if what we get in the arms is compatible, or at least can to some way capture some of the biodiversity within the rhodolith. So it must be tested before, as we did with the coral reefs. But yeah, you could use arms there. And as to the anchoring thing, you can use weights, and you can just use a concrete block or weights to anchor in place and leave it there. It has been used in different spots.
Okay, thank you, Tito. Okay, we move on. Rafa, there is a question for you. It's in the chat. How much clear or evident is the source or cause of our environment problems? The biodiversity crisis, climate change, to coastal management tools developer. How to address that cause? Going deeper, how to provide effective conservation, consider the logic necessary linear economic growth based on consumption that domain the global policy? Hard question.
Yeah, this this is a tremendous question. I would say it's a question for one entire course. We live in a production consumption global machine. We live in a world in which is moved with this production consumption global machine, and now we know, we have the evidence that if we change the way in which we produce and we change the way in which we consume, we have no future. We know that. We know that. And also the problem of the COVID is showing us that the world needs to change much faster than it was expected. All these crises that these people, this person says, are due basically to two issues. One is that we have more population. In the last 12 years, I think it was in the last 12 years, we increased the size population of humans in 1 billion people. So people is saying that in the 2050 we will be 10 billion, but I think there will be more. And we cannot do exactly the same that we used to do before with more and more people. When I born we were 3 billion. Now we are 7.7 billion. So people. And the second one is activities, and it's what it was stated in my second slide. We need to grow. This is something that is required to grow, and we grow 3% globally. And if you put these two growths of population and activities, you have the scale of economy, and the scale of the economy is always growing. And because those are two growth factors, this growth is an exponential curve. So if you put all these problems that this person was saying at the beginning, climate change, biodiversity, and we manage all these problems exactly the same that we did it in the last 30 years, we have no solution at all. So we can accept more people, we can accept more activities if we change the way we do things. We need to produce differently, and we need to consume differently. And this is the challenge that we have as humankind for the coming decades. In which way we need to change rapidly the way we do things. And for us, for me, which I'm just a very old guy, it's really difficult. But for young people, that has the entire life in front of them, these guys needs to be the drivers of change. So I will encourage whatever of these people that are that's hearing us these days to be an ambassador of change. To do not conform it with the thing that says that we will be able to deal with that. We need to deal with that right now by changing the way we do things. And it's very easy to say it and very difficult to do it. But I think that right now there are several businessmen that are just moving in that part. There is a conference on Reuters next week with big CEOs of companies and big presidents or directors of NGOs here in Europe, and it's about this. It's about how to make a much better sustainable path for the future. But basically, we need to change the way we produce, the way we consume, and to solve all climate change is clear, but biodiversity is also clear. We eat too much meat, and well, as I told you, it's very difficult to answer a question like that, because it will be just one entire course just to show some path, some maybe path in order to redress these things.
Good. Okay. Very nice.
But by the way, I saw 1 week ago to the man or lady that says this question. I saw one week ago a fantastic documentary in TV of David Attenborough. I don't know if you know him, and this guy is right now making fantastic docs. And in this documentary, he was just saying the way in which you should change. And if you try to find this, it's very, very good. It's very, very good.
Okay. Thank you, Rafa. Guys.
I'd just like to make a brief comment, just because, yeah, we were seeing a lot of awareness raising in the past, like 100 years. People become more and more aware of the environment and the problems that we are facing, and now we face this urgent problem. It's just enormous. It's right there. We're going through the warmest period in the 100 years, and at the same time, in the past like 4 or 5 years, the whole world and important economies and players in the world have sat back to wrong political choices and electing people that don't believe on these problems or disregard all these things, like in the US and here in Brazil. So this is sort of mind-boggling, because I couldn't understand how we were coming from raising awareness constantly and informing people and making them more sensible to these questions. At the same time, the whole world is turning to the wrong direction, with people directing the planet towards destruction. So we have to, to start informing people more strongly, and also informing about the political consequences and the environmental consequences of the political choices. That's a big thing too.
Okay, very good comments.
I don't know in Brazil, but I hope, I'm crossing fingers, that in the States in 1 month they kick in the ass the guy that they have right now. And I know that there are a lot of young people that are right now racing the boats in order to kick out this guy.
Hopefully. Fingers crossed as well. Rafa, keeping talking about issues, okay? You have talked about this very interesting too, but I face the question for you. But what do you think is the biggest barrier to have the ecosystem-based management applied for a place? I know that's like different place you have different issues, but if you look worldwide, what do you think is the biggest barrier?
I don't get the question. The
What is
biggest
barrier? Barrier for
barrier. Okay.
yeah, sorry. For
Well,
have this tool working.
I think that the ecosystem approach is not well taught in the schools, in the universities, and there are a lot of different ways to teach this. And when you go into practice, you end up with the idea that many of the people that they need to apply that, they don't know how to do it because they are not formed on that. So the ecosystem approach is a down, I would say, it's a down strategy. So it came into universities probably just 10 years ago or less. So there have been very few scholars that has been teach on that. And when you go into practice, many of the people that are still doing managing these things are all guys that were not, they don't know how to do it. I think that there is a barrier in capacity building here, and everyone we did in the past we did a master thesis of one lady that went into different marine parks here in Spain and was making interviews with the managers, and at the end the responses of these managers were completely different. Everyone was understanding the application of the ecosystem approach in his own vision, and everyone was fine. So everyone thought that he was doing the job, but you could not put these guys to work because they didn't understand each other, what they were doing in relation with the ecosystem approach. So there is a big difficulty to introduce these principles into real management in a normative way, I would say. So I don't know if you can say a barrier, but the problem is that there's a confusing way in which you apply the ecosystem approach in every particular application.
Okay. Thank you, Rafael. You answered my question. Okay, we have another question for Genuario. Genuario, that's from Sergio Rossi. Some of the Cysts may have an internal chronological clock to restart depending on the season. How much do you know about that?
Yes, they have an internal clock. This is the problem of the biorhythms in general. But the internal clock, it doesn't depend from the environment. So it's simply switched on, is a timer, is switched on by the mother, in the case of my animals, the copepods. So is the mother who switches on the timer of the eggs. For example, the female of September produce eggs which will hatch in March, okay? But the females of the same population, of the same lake, and of the same situation, females of December will produce eggs that will hatch in March. So it's not a problem of 6 months. It's a problem of perception from the mother of what is the calendar day in that moment. And the mother is able to switch on for a certain period the timer inside the eggs. The eggs can perceive when they hatch, can even perceive that it's still not the moment, it's not the proper moment. So they can re-enter in another loop of resting. This is why when a resting egg, a cyst, is buried in the sediment, it can rest for years because it doesn't perceive the suitable condition when he is in the moment of the hatch. So there are two different physiological phenomena. The diapause is the genetic programmed rest, and the quiescence. The quiescence is a rest against unfavorable conditions. Okay, they are not the same thing, but they can add each other to produce this long time rest, and this travels not in the space but in the time. This is incredible, but what we are searching to obtain with technology and with our studies, in nature it exists. So in nature, an organism, an individual can travel for 100 years in the future with this mechanism.
Okay. Thank you. Nice questions. And yes, that's a personal question. Like from what I understood, you also have these environmental keys for hatching. And we know that, for example, the ocean is getting warmer. How do you see, this changing of the environment, changing how these cysts hatch? Because, for example, if a plankton species needs a 20 degrees temperature, and because the ocean is warmer, you don't have 20 degrees every year.
Yes, we can have consequences, sure. But this is a small problem. The larger problem of linked to the cyst production is that of the ice cap, for example, in the North Pole. Because when we will have not any ice during winter, we will lose the possibility for the phytoplankton during summer to produce biomass. This is terrible. So without ice, we will not refuel the water with cysts and with active stages of phytoplankton, and the phytoplankton will disappear from the North Pole. With all we cannot imagine the consequences of this, but this is a very large consequence, not simply for each species at the variety of 1-2 degrees or more. This is small problems for each species. The global problem for all the planet is that of the melting of the ice cap in the North Pole.
Beautiful. Yeah, nice. That's another issue for the Arctic and for the poles. Yeah,
it's an scary issue.
For the the entire ocean. Okay, Tito, I have a personal question as well. Yeah, it's more or less about the global change. We always talk about bleaching when you talk about like coral reefs, but from your talk, I understood that cryptic are very important as well. What do you think is the biggest risk for this kind of fauna nowadays? What?
Well, the biggest risk is the total collapse of the system, because if the bleaching is like the canary in the coal mine, it's like signaling what's happening with a very sensitive organism, but still we don't know what's happening inside the reef matrix that could like facilitate the bleaching process. We know that there are some organisms in the matrix that may help the coral to withstand the temperature going over the normal temperatures in the coral reef. So like they have done some research with polychaetes, like the spirobranchs, that around the burrows of the polychaetes, the coral tissue is healthy, while the rest of the coral is bleached. So there's some interactions with the organisms that may help the system to, to at least cushion a little bit the effect of the heat waves in the system. But on the other hand, we don't know what the heat waves are doing in the cryptic fauna. So that's the data we are generating now with the arms deployed in different reefs around the globe. So we are processing this data and figuring what's happening, if we can see any changes related with heat waves. Because what you see in the arms is a change that has been accumulated over the period of 1 year or 2 years, because this time you leave them sitting in the bottom. So you cannot see like a snapshot of what's happening in the moment of the heat wave. So you get what happened during that event in a broader, time scale.
Nice. But if you compare like two different years, you can see the difference. Like,
yeah, because as I said, there are many animals that rely on each other. If the coral dies, what's happening with the crabs that lives within the corals? They will disappear, and the coral will be covered with algae or blue-green algae, bacterial biofilms, and this is not the same environment for these guys. So all things are linked into a complex food web. When you take some of the key elements of this food web, you can change the whole thing, including the cryptic fauna.
Beautiful, beautiful. Any comments, guys? Rafa, Genuario?
Well, I guess that it will not be so difficult to make an experiment with arms in same year in places in which you presume that it's going to be bleaching and areas or not. I was talking about the documentaries before. Have you seen this Chasing Coral documentary? And if you see the Chasing Coral documentary, these guys may have the knowledge about which places can be much more possible under bleaching, and you can just set up arms at the same moment, and to see evolution in relation with that. It has been done before. Is kind of experiment like this one.
I think they are doing this. I'm not really aware of which group is doing this sort of experiments, but there are many groups using arms around the globe and doing different, even manipulative experiments to see what's happening. There are many ways you can use the tool. Yeah, but it's very interesting. Yeah, to check what's happening right on at the same time that the bleaching event is occurring. But I believe that most of the effects will be long-term, will be after, the coral dies, because the bleaching is a very acute thing. So the coral will put away the dinoflagellates, but during that time, most of the tissue is still alive. It will die off depending on the place you are. In the Pacific, they will come back easier than in the Atlantic. But the coral is still living during the bleaching event. And after the bleaching, they may die if they will not recover the dinoflagellates. And then the process, it's a little bit slower. It will progress into, to be covered with filamentous algae and all other things, and then we'll progress into a different. It will go through what we phase shift completely.
One thing, one comment, because bleaching is something that normally I'm a little bit familiar with that. But the issue you say, Genuario, about the phytoplankton and disappearance of the phytoplankton related with the loss of ice in the Arctic. Can you explain a little bit more, because this is something that you scared me, and what is the reason?
Yes, so trying to put order. So if we have not ice in Northern Pole during winter, we have not cysts entrapped and during the night of the winter of the Northern Pole, because we have not sun for 6 months. So if the ice is not produced, we have not the possibility to trap inside the ice in the sympagic space, also the phytoplankton in terms of cysts. At the successive summer, when the ice melts, all the cysts are made free and the phytoplankton is reproposed. But if we stop with the ice formation, we will have not phytoplankton at all.
Okay.
This is a complex scenario, but if we will have the time and the possibility to perceive perfectly this possibility, because it is an hypothesis at the moment. Many Finnish colleagues, many Norwegian colleagues, they find animals in the sympagic space. So in the sympagic space, many organisms are present, but they hatch. They are born from the hatching of eggs and of cysts, or they still stay normally in the sympagic space. They are the representation of the plankton, or they are typical of sympagic spaces. So all these things we have to understand. But the possible scenario could be this one: the disappearance of plankton produced by the disappearance of the ice. Because the ice is the only possibility for the phytoplankton to survive during the night, the Northern Pole. Okay? If the phytoplankton doesn't survive, the year after, we will have not phytoplankton. Simply.
My God.
No.
Okay?
Yeah,
It's too much simple, okay, but it's terrible.
It's very scary. Any comments, guys? Rafa, Tito? Okay. We have a question for Tito that's from Paulo Horta. Tito, with the advance of molecular big data, we are facing that evolution is a continuum process. How much this knowledge strikes the binomial linear perspective of our biodiversity? And he has a comment. In my perspective, the binomial segregation of our biodiversity produces a false perspective of evolution that isolates species, mistaking evolutionary interactions and interdependencies. In different places of the world, we have different amounts of information about. No, that's another topic.
Yeah, well, if I got right the question, yeah, what you're seeing in terms of our understanding of evolution have also evolved a lot. So, and I agree in terms that when we think of species as an isolated entity, that it's not evolutionarily related with any other things, or it may be only shared some common ancestor in the past. Yeah, we are seeing that evolution is not operating on this way. We can, especially if we think of the diversity of different organisms like bacteria, plant, animals, algae, all sorts of microorganisms, archaeas. they all evolve in different ways, and they exchange genetic material within them, and these organisms can also operate exchanging genetic material within different organisms, like animals and plants and things like that. So yeah, we have now a different understanding of how evolution may operate, and we can see that it's not really each species in its own lineage just evolving alone and everything. Yeah, it has changed. At the same time, if you put the Linnean binomial system of naming things into the same basket, it's a different thing, right? Because in a way, it helps us to establish a common language, so everyone knows what we are talking about. So this sort of stability that is important in taxonomy, it has kept the science, progressing. But I know that it's also a hurdle for in some way. So it's been discussed in all the forums, the problem in the Linnean system of organizing the biodiversity information. So this is slowly changing, and I believe it will absorb all these aspects of the evolutionary processes that we know more about and all the other things. I don't know if I really answered the question, but that's my thoughts.
Okay, I think you answered that. Yeah, no more questions here. Rafa, I have last questions for you, okay? And for example, in different places of the world, you have different amount of data about the ecosystems, okay?
Mm-hmm.
Something is talking about coral reef in Europe, and something different is talking about a coral reef system here in Brazil or Africa, whatever. One of the base of your of the ecosystem-based management is data.
No.
So what's the work around this issue?
You need to work with what you have. So if you have less data, you need to work with less data. It depends. It's clear. Not all the places in the world will have the same amount of information. Probably in places in which you do not have a tremendous amount of information, but you know that the area is important, you work much more on species based. So the information is based on charismatic species or key species or just habitats, and you work with this type of information. In other places in which the information is much higher, then you can develop other aspects about biodiversity or about other things. Of course, you will never work with the same amount of data. That's clear. And this is not an impediment in order to protect an habitat if you think that is important.
Okay, beautiful, good question. Any comments, guys? Okay, so we don't have any more questions. So I will ask you for final comments. We can start with Genuario. Final comments for our morning.
Final comments. Really interesting, this exchange of opinion and of positions between colleagues who work in different fields, evidently. So the biomolecular approach, the explorative approach is mine, the ecological, so the synthesis of the approach of Rafael. So for me, it's very useful and productive to stay here with you today. I hope that this could be also for the people who attended this these 2 hours, 2 or more hours. So,
Yes.
and we obviously, I'm available for any question for all the people at my email if they have. So it's very interesting, and I would like to prosecute this kind of exchanges, even in the immediate future.
Yes. Yeah, thank you, Genuario, for your words. I personally think the coronavirus brought something good. It's the opportunity to have this kind of talks. You may not have this kind of talks if you're sometimes we don't have the money to bring like 9 different people to Brazil or to Europe, and online you are doing that. Yeah, that's important. Rafa, final comment.
Final comment. I will come back to my first slide probably, and I don't know how many people is seeing us this afternoon, but to all people that is just seeing us, I will recommend strongly to read these 2 papers that I said at the very beginning. The one about Carlos Duarte, Rebuilding Marine Life, Nature, this year. It was around March or February, March this year. A fantastic, very positive paper about the possibilities to recover marine life. And on the other side, the other one, which is based in one paper, the paper is called Blue Acceleration, and it's in a not very known journal called One Earth Perspective. But I will send you the paper if you have connection with the guys. This is a fantastic paper in order to pursue the fact that the demand for food, materials, and space in the sea is going to increase heavily during the next decades. So you have this dilemma. On one side, you need to preserve the wildness. On the other side, there are demand for this wildness. We're talking about the marine domain. The terrestrial issue is we are doing late, but in the marine domain, we have this dilemma right now. Scientists demands people to put a number of miles of protection in the sea. 30% of the entire sea water should be protected in a near future. United Nations give us the target to protect 10% by 2020. We didn't do it. It's the same like with climate change. The targets are never do it. And I think that for young people, and this is my comment, my general comment, and my general message, especially for young people, they need to work heavily that those targets should be accomplished in the future and in the near future. We know the problems. We know the problems where we are. We know in which kind of life we are going to live in the future if we don't solve these problems. So we need to work heavily to deal with these issues. And well, my general comment is, okay, we need to protect more space in the sea, and then if we protect the space, then we can manage the space. So my talk was about to manage something that has been protected. So, but first we need to protect. And in order to protect this space, we need to put pressure on these policy guys that Tito was referring before to let us understand them that this protection is basic for the life of the entire humanity. So, well, we need to speed up a little bit our response in the demands that we have in front of us.
Beautiful words and very important, Rafael. We have people here asking you to send us the reference for the 2 papers. Okay.
Yeah, sure.
Okay. Thank you.
So I sent to you the presentation because we were engaged in this problem that I couldn't put the presentation. I will resend the presentation with the literature, and I didn't realize that I sent the presentation to you with some pictures at the end that are not in the presentation. And there is one picture of a friend of mine and his wife try to get out of this picture before whatever.
Okay. Okay, well, thank you.
Sorry about that. I didn't realize that.
Sorry, yeah. Yeah. We did a lot of tests last week. Everything worked, but today didn't work. We don't know why. That's it. Tito, final comments.
Oh, well, thanks a lot. It was I learned a lot during this morning. Well, morning for us here in Brazil, at least. So thank you, Rafael and Genuario. It was really, really interesting, different perspectives. Really looking forward to read all the papers related to your work later and understand more and more about the issues you're raising here. Well, as my final comment, I would like to to make calls with Rafael also about the urgency of preserving our biodiversity. I think I try to make it clear that when we assess the biodiversity using different tools and even like molecular tools like Metabarcoding, what we see is a huge fraction of that information that is still on the gray, we don't know what it is. So it's still a lot to do in terms of knowing the biodiversity. So this tool is detecting the amount of ignorance we still have about what we have in the sea, and we are losing this biodiversity day by day. I really hope that in the future, using different tools and different methods to assess biodiversity and monitor biodiversity, we could see a change in a positive way. See this coming back to what it was, maybe, but it's a hard task. We have to still promote the studies on the biodiversity still, and the problem of that Rafael raised about hiring people for doing the basic taxonomy and to do the ground and the basic information that we need to know what we want to preserve. It's essential. So we have to change this policies, and we have to change the way we are also communicating our science and making people more and more aware of what's going on, and try to change the course of this big ship.
Yeah.
Thanks a lot.
Yeah. Okay. Thank you, Tito, and yeah, thank you everybody for being here. Rafael, Genuario, Tito, very good talks. I love the questions, and thank you. We have around 100 people watching us now, but yesterday you had thousands, really thousands of access to the YouTube video. So a lot of people, you'll be hearing what you guys just said. So basically, we're gonna close this morning section. We'll be back this afternoon, and yeah, thank you guys again, and yeah, see you in the future. Bye bye.
Bye bye.